Okay, everybody, we have one last test to learn this semester. It's called the Kruskal Wallis test, named for its inventors. And it is the non-parametric test that we're going to use when we want to look for a difference in means, like we would in the same kind of experimental design that we would when we're doing an ANOVA. So more than two groups where we want to look at means. However, we violate the assumptions for ANOVA, or we're interested for some reason in looking at a difference in medians in the first place. All right, so this is an experimental design. It's the same as we would use for ANOVA, but we violate the ANOVA assumptions. It's going to be a little different because, like the rank sum test, it's going to be a test on medians, not on means. And we're going to write the null hypothesis to reflect that. The assumptions for ANOVA are that the data are normally distributed, that the standard deviations of each of the groups are approximately equal. And also, ANOVA works best if the sample sizes between the different treatments are close to equal. So if any or all of these assumptions are violated, then we need to switch from doing our planned analysis of variance to doing a Kruskal Wallis test. All right, so we're going to write our null hypothesis. Now, normally when we're writing our null hypothesis about means, we're going to use the Greek letter mu to represent the means of the population. We don't have an equivalent notation that we're going to use for median. So we're just going to write median out, and we're going to use subscripts like we do. If we have a letter that we can substitute in that helps us remember what our different groups are, our treatment groups, then we're going to use that. So our null hypothesis is always going to be the medians are all equal, starting with the treatment group number 1. And going through capital I. So this letter might look like 1, oops, but it's a capital I. All right. Our alternate hypothesis is that the medians are not equal, and so we're going to use those not equal signs in each of the steps in between them. And it's similar to the rank sum test, but we need to find the ranks for all the groups instead of just looking for the one with the lowest sum of ranks in order to calculate the test statistic. So just like all of our other hypothesis tests, we're going to have our null and alternate hypothesis. We're going to calculate a test statistic like little t, like capital F, like little z, or chi-squared. We're going to calculate this test statistic. We're going to call it capital H. Capital H has kind of a strange looking equation. It's a little awkward. I can't explain to you how it's derived or where all these different values come from. Somebody figured it out, but it's not obvious. So we're going to take 12 divided by capital N times capital N plus 1. So capital N is going to be our total sample size, however many total individuals summed up across all our different treatment groups. Each treatment group is going to have a sample size. We're going to call those little n sub i's. So our first treatment group is going to be little n sub 1. Little n sub 2, our second treatment group. It's nice if they're all the same as one another, but they don't have to be. So little n sub i's could be different than one another. Again, capital I is the number of groups, and then we're going to calculate this value, or we're going to find this value called capital W sub I. That's the sum of the ranks of the ith sample. So just like we found the ranks when we're doing a rank sum test, we're going to find the ranks out of the whole data set mixed together. And then we're going to sum up the ranks within each of the sample groups. Those are going to be our capital W sub i's. This formula tells us to take the square of each of those sums of ranks and then divide it by the sample size for that group. And we're going to multiply it times this quantity that's based on the sample size. And then we're going to subtract off this little caboose of 3 times capital N plus 1. This quantity, capital H, is distributed approximately the same as the chi-squared distribution. So even though we're going to calculate a capital H, then we're going to go to this chi-squared table in order to find our p-value for this test in order to compare it to our alpha like we always do. In order to use the chi-squared distribution table, we need to have a degrees of freedom, and the degrees of freedom is going to be based on the number of groups that we have. So it's going to be capital I minus 1. If we have 3 different groups in our experiment, that means our degrees of freedom will be 2. All right, so make sure you remember that and have that written down in your notes. All right, we're going to do an example to work through how you do this test, because finding capital H is more of a set of steps. It isn't just doing a calculation. So we have different colored sticky cards that were set out in a field, and we wanted to see which color, how the different colors trapped different kinds of insects. So we had 6 different sticky cards in each of these 4 colors, and we left them out in the field, and we counted up the number of insects that were stuck on them. All right, so these data are not normally distributed. We want to know, is there a difference in the median number of insects on the different cards? So we need to write out the hypotheses for our test. Our null hypothesis is going to be median of yellow equals median white equals median green equals median blue. Our alternate hypothesis is that the medians are all unequal. Now we have 4 different groups. So our capital I is going to be 4. Our little n sub i's are all equal, and they're equal to 6. We have 4 groups of 6, so our capital N is going to be 24. So we have the different values that we're going to need to plug into our equation for capital H. Now we have to find the ranks and sum them up. All right, so remember that we start with the smallest value in our whole data set, and we assign it rank number 1. So here we have number 7, and we've assigned it rank number 1. Then we go to the next largest value in our data set and assign it the rank number 2. We're going to keep looking. The next one is 12, so we'll assign that a rank of 3. And then we find a 13, so we assign that a rank of 4, and we find a 14. All right, now we would assign it a rank of 5. However, there's another 14 in our data set. All right, so these 2 14s, we want to assign them the same rank. However, we have 2 ranks that we need to use up, right? So we need to use up the rank 5 and the rank 6, but we need to do it in a way that assigns both of these values the same rank. So we're going to take the average of the rank of 5 and of 6 that we need to assign, and then we're going to assign it to each of these separate values of 14. So when we have a tie, we have a tie between observations. It doesn't matter if they're in different groups or if they're in the same group. You could have 3 or 4 values that are all tied together, right? We still want to take the average of all the ranks that should be assigned to the next number, however, of observations it is. We're going to take the average of all of those ranks and assign the same rank to all of those tied observations. All right, so we end up assigning a rank of 5.5 to both of these observations that are the value 14. Now we're going to keep assigning our ranks. So we would have assigned a rank of 5 here and then a rank of 6. So the next one we have to assign is a rank of 7. And that is going to go to the next value, here 15, and then the 8 goes to observation valued at 16. And then again, we have this tie for the 2 data points that are both 17. So they should get, we have 2 data points, we need to assign 2 ranks. So they should get the ranks of 9 and 10, right? But we're going to assign them each the mean of the values 9 and 10. So they each get a 9.5, right? So we should have assigned the ranks 9 and 10. So the next rank that we have to assign is rank 11. So rank 11 is going to get assigned to the value 20 because it's the next one, right? So 21, we have 2 replicates of it. So again, we have to assign the rank for the ties and keep going through the whole data set, all right? So double check and make sure that you are understanding how to assign the ranks across the whole data set and that you've done it correctly working out this example for yourself. All right, so once we have, if we've written it out this way, then we just are going to sum up the ranks of each of the colors. So we just add those together and we get the capital W sub Y for the first group, the little I, we're going to call it Y. Little I sub 1 equals 127. Same thing, we're going to sum up these ranks. And we get 44 for white. We sum up the ranks, right? The ranks, not the data, right? Make sure you're summing the ranks that you assigned. We get 89. And then for blue, we're going to get 40. Right, so those are our W sub I's. And now we're going to put all of the data that we have into the equation for capital H. 12 divided by capital N, which is 24, times 24 plus 1. We're going to take each of the values of capital W and square it and divide by the sample size for its group. They're all 6 all the way across. And then we're going to subtract off this little caboose, 3 times capital N plus 1. And when you're working through this equation, and it's kind of long and complicated over here, don't forget to subtract off this last little bit. All right, so we get our value of capital H equals 16.95. Well, what the heck does that mean? What do we have to do next? We have found our value for our test statistic, so we need to find the p-value associated with it. Remember, for Kruskal Wallis, we're going to use the chi-square distribution. And in order to use the chi-square distribution, we need to know the degrees of freedom. Right, so the degrees of freedom for our Kruskal Wallis test is going to be capital I minus 1. So we've got 4 groups minus 1, that's going to give us 3 for our degrees of freedom. We're going to go to that row on our chi-squared distribution table. We'll see that our H is in between the values of 17.73 in the table and 16.27 in the table. So we're going to go to the top of the table and find the p-values associated with those values of chi-square. And we find that it's in between 0.001 and 0.0005, right? If we have an alpha of 0.05, is our p less than our alpha? Yes, our value of p is less than 0.001. That means it's less than 0.05. All right, we're going to reject our null hypothesis. So if you scroll back up to the top, you'll see that our null hypothesis was that the medians were all equal from each of the different groups. So if we reject that null, then we can say there is a statistically significant difference in the median numbers of insects on the different colored cards. Now, if you're curious and you go back and look at the data, think about if you wanted to trap a lot of insects, what color card would you use? So you want to inspect the data, kind of see informally which one is the one that had the highest or the lowest or whatever you're interested in. All right, so the only tricky parts of a Kruskal Wallis test are remembering that you need to use the chi-square distribution and also doing the assignment of the ranks. Right, the assignment of the ranks, sometimes you just make a mistake. It's a little tedious to do. All right, so we've got one more practice problem for you to try. It's a similar question. We've got 6 different field sites. They had different types of substrate. All right, so substrate is what the organisms are sitting on, and it's either rough, smooth, or mixed. Right, so we might have uneven surfaces, smooth surfaces, and then a patchy distribution of each. So we've counted up invertebrates on these different substrate types, and you have the counts here. These are appropriate data for a Kruskal Wallis test. They're not normally distributed. Right, so pause the video here and write down these data, and then try and assign the ranks and find your summed W's, and then calculate the value of the H statistic. I'm going to show you the answer so that you can check your work, right? But if you don't want to see the answer yet, then pause the video and then you can compare and see. So there's a couple more practice problems in a separate file that you can find in the module that has the answers included. So make sure you do a couple practice problems so that you're calculating the capital H test statistic correctly. So here is the work that I did finding the ranks. Right, notice I made a few mistakes and realized I hadn't gotten them all. So if you have 6 times 3 observations, you should be assigning ranks up to 18. So if you get to the end of assigning your ranks and you've either assigned too many or too few from what you were expecting, you can tell that you made a mistake. Something I do that helps me make sure I'm correcting my ties is that I sort of pretend to assign the 2 ranks that I need to. So for example, when I got to these tied observations, I actually wrote down 5 and 6 here. I should be assigning the ranks 5 and 6, but I'm going to assign 5.5 to each of these observations. Now I can remember more easily which rank I need to go to next. I should have assigned a 6, so I'm going to go to a 7 next. Right, so you can double check you did that correctly, that you got your W sub i's added up correctly. This is the first time I've worked this problem. If you notice you're getting a different answer, shoot me an email and let me know in case I made a mistake. That's always possible. Once you've gotten those done correctly, you should be able to work through calculating capital H. And here is what you should find. All right. All the other steps of doing the hypothesis test are exactly the same as we've been working through, finding the p-value, deciding to reject or accept the null hypothesis. All right, that's it. You've got the Kruskal Wallis test in your toolbox, and that's all there is to it.